Yes, these are my co-workers. This is from Europe, this is from China, and these are from Russia. Yes, next please. As I told you that Alzheimer disease is a very devastating disease, and so far there is no cure to treat this disease. Therefore, we have to try different models and different type of strategies to treat them. Next please. As you can see that mild cognitive impairment, the ventricles in the brain are enlarged, and in Alzheimer disease, most of the tissues are lost at the expense of ventricles. Next please. This is a cartoon showing that in normal condition, and this is mild Alzheimer disease, the ventricles are enlarged at the expense of brain tissue. Here is the severe Alzheimer disease. The brain tissues are much more lost at the expense of ventricles. Next slide, please. These are some examples that military risk factors for Alzheimer dementia and neurodegenerative diseases. Next, please. Military risk factors for Alzheimer disease. They are well discussed in this paper, Alzheimer and dementia. Next, please. Amyloid pathology fingerprint from different post-traumatic stress disorder and traumatic brain injury. There are three different things: traumatic brain injury alone, post-traumatic stress disorder, and post-traumatic stress disorder with traumatic brain injury. I will show you some images. Next, please. You can see here these light yellow is a reflection of amyloid beta peptide in normal. Here is in the brain injury, traumatic brain injury. Then here is post-traumatic stress disorder and the combination of traumatic brain injury. So you can see that the combination in post-traumatic stress disorder and traumatic brain injury is the maximum. Next slide, please. As you can see that some amyloid beta peptide deposit in blue, they are normal. But here in brain injury, they are much more increased and abnormal. And this is the post-traumatic stress disorder. They have the maximum number of amyloid beta deposit in the brain, human brain. Next, please. As I told you and also showed you previously in animal model, various regions are different. Here is frontal, parietal, occipital, cingulate, and temporal cortex. As you can see, that PTSD, post-traumatic stress disorder, is highest among all cases. Wherever different regions have shown different kind of increase in amyloid beta peptide. Next, please. As we are telling always that blood-brain barrier is one of the key factors in all pathological diseases, there were some questions that can blood factors affect Alzheimer disease, and this is one of the classical paper in National Academy of Proceedings of National Academy of Sciences in US. One of the prestigious magazines said, blood-derived plasminogen derives brain inflammation and plaque deposition in a mouse model of Alzheimer disease, means plasma factors affecting brain function. Next, please. Our theory for blood-brain barrier disruption is perfectly proved by this example already from Netherlands, that mild cognitive disturbances showed leakage of blood-brain barrier in human brain. Next, please. We used a genetic model of Alzheimer disease in mice and measured leakage of albumin. You can see much more expressed here. But when we treated the cerebrolysin, this is rather much reduced or absent. This suggests that leakage of albumin in the brain has important contribution to the development of Alzheimer disease model. Next, please. These are the same I showed you that nanowired cerebrolysin has significantly reduced amyloid beta in the brain, in the cortex. Next, please. This also happened with p-tau. We are labeling nanowired cerebrolysin, and it has significantly reduced the upregulation of tau protein. Next slide, please. These are the methods. We are measuring blood-brain barrier, edema formation, neuronal, glial, and axonal injuries. I will show you in animal model. Next, please. We have developed a model of concussive head injury by dropping 114.6 gram weight over the exposed parietal bone from a 20 centimeter height that produces an impact of 0.224 Newton over the skull surface. That is called mild traumatic brain injury. Next, please. Then we have administered mesenchymal stem cells, nanowired, and then we have used cerebrolysin, and then together TiO2 nanowired cerebrolysin with mesenchymal stem cells, and see the result. Next, please. This is the model of head injury in rats. This is the weight dropped over here, produces brain injury, no skull fracture. Next slide, please. In this model, right side is injury, but as in clinical cases, due to counter-coup mechanism, left side showed much more advanced brain damage that is supported by clinical cases, and also we found in animal cases. Next slide, please. These are the US military data that various kinds of severe, moderate, mild, and unclassified brain injury is a real problem. This is the case of 244,217 cases in US up to 2000. Next slide, please. Without concussion, there is small network problem in memory and motor planning. But after concussion, the neuronal network for working memory and motor planning is severely damaged, as you can see here. Next slide, please. Then I show you the example of amyloid model with CHI and measure various functions at histopathological conditions. Next, please. Therefore, we suggest that nanotechnology is the future for therapeutic strategy in various neurological diseases. This article appears in Future Medicine. Next slide, please. We have examined single therapy and then combined therapy in both concussive head injury and Alzheimer disease model. Next, please. These are the nanowires where the drugs are labeled. This is a US patent technology. Next slide, please. This is Dr. Wang, and this is Dr. Ryan Tian in University of Arkansas Fayetteville, United States of America. They are the patent holder, and we are working together with them. Here I can show you that even stem cells, when nanowired, they are long-lasting and much more effective than the normal or conventional stem cells as treated in various diseases now. Next slide, please. This is an example that traumatic brain injury exacerbates amyloid beta peptide-induced brain pathology. Here is blood-brain barrier to iodine, brain edema by water content, Alzheimer disease pathology by neuronal injury, ABP deposition, and number of albumin cells. These are saline infusion. Then ABP infusion has significantly increased in all conditions. TBI means traumatic brain injury or concussive head injury plus AD, much more significantly increased. Nano delivery of cerebrolysin, they are reduced. Nano delivery of mesenchymal stem cells, they are also reduced. But when we combine both nano delivery of mesenchymal stem cells plus cerebrolysin, the values are significantly reduced in all parameters we have examined. Next slide, please. This is an example of nerve cells showing untreated. Many nerve cells are gone, widespread edema. The nanowired mesenchymal stem cells, the picture is much more better than this picture. Nanowired cerebrolysin, you can see the picture is somewhat better than this one. We combined here nanowired cerebrolysin and nanowired mesenchymal stem cells. The nerve cells look near normal in the neuropil. Next slide, please. This is a diagram of neprilysin. That is, when it is decreased, there is more brain pathology. Next slide, please. You can see that neprilysin is associated with astrocytes, and there are different diseases. You can see the absence of neprilysin and also the astrocytes. Next slide, please. This is a table for different parameters are measured: leakage of blood-brain barrier to Evans blue and iodine, brain water content and volume swelling, ABP deposition, albumin deposition and neural injury. Normal regions, the values are very less. TBI, the value has significantly increased. But ABP infusion, also the values are increased. And when we have used nanowired cerebrolysin plus ABP infusion, you can see that after TBI, all values are significantly increased. When we have used nanowired cerebrolysin plus nanowired neprilysin, then the values in normal and TBI significantly decreased. Next slide, please. This is an example in hippocampus. Traumatic brain injury plus Alzheimer disease, very few cells can be seen in hippocampus, whereas treated with cerebrolysin, nanowired cerebrolysin, cells have significantly increased, not died. Combination of cerebrolysin and neprilysin in same conditions has significantly protected many nerve cells near normal values. Next slide, please. Another important point is stress. So we used resistant stress and Alzheimer disease pathology and found that stress significantly increases the pathology of Alzheimer disease. That's why we have seen post-traumatic stress disorder exacerbates brain pathology. Since immobilization stress induces breakdown of the blood-brain barrier, we have shown for the first time in 1980s. It is now proven by various other observers that this is true. Next slide, please. This is an example of amyloid beta significantly increased Alzheimer disease plus resistant stress. When we have given the drug treatment, nanowired cerebrolysin plus monoclonal antibodies to ABP and tau, these are largely absent. Next slide, please. Cell damage, you can see untreated group and the drug treated group appears significantly different. These pictures, the large, dark and condensed neurons were largely absent in this image. Next slide, please. This is EVA1, one microglia representing. Untreated, you can see upregulation of microglia can be seen here, but they are significantly reduced by combined treatment of nanowired cerebrolysin with amyloid beta peptide and tau antibodies. Next slide, please. This is CD86, another kind of microglia evaluation. In untreated group, you can see upregulation of CD86 is severe, whereas this is significantly reduced by the combined treatment using nanowire technology. Next slide, please. At the electron microscopy, we can see the damage to myelinated axons and edematic formation here. And here you can see the treatment. The axon is better preserved and only few edematic cells can be seen here. Next slide, please. We have seen that nanowired mesenchymal stem cells, when combined with cerebrolysin, they are producing significant neuroprotection in cases of Alzheimer disease with brain injury. Next slide, please. This is amyloid beta peptide in parietal cerebral cortex. Concussive head injury with Alzheimer disease, much more significant here. Nanowired cerebrolysin alone reduced markedly. Nanowired cerebrolysin plus mesenchymal stem cells and antibodies to nitric oxide synthase further enhanced neuroprotection in this case with regard to amyloid beta peptide deposition. Next slide, please. Toluidine blue in parietal cortex, brain injury alone, brain injury plus Alzheimer disease, you can see many neurons were damaged. Nanowired cerebrolysin has rescued many of them, but the nanowired cerebrolysin together with mesenchymal stem cell and antibodies to neuronal nitric oxide synthase significantly enhanced neuroprotection. Next slide, please. NeuN, that is the marker of neuroprotection, is severely, largely absent here in CHI. And furthermore, this is also deterioration can be seen in NeuN upregulation in combination with Alzheimer disease. Nanowired cerebrolysin here, many neurons were looking normal, but the combination with nanowired cerebrolysin with mesenchymal stem cells and antibodies to neuronal nitric oxide synthase has significantly enhanced neuroprotection. Next slide, please. CD86 again, CHI alone, combination of CHI with AD, nanowired cerebrolysin alone, and nanowired cerebrolysin with combined stem cells and antibodies to nitric oxide synthase, done little better than nanowired cerebrolysin alone. Next slide, please. Vimentin during development is present, but when disease occurs, it can reappear. As you can see here, that in CHI, some vimentin is upregulated here in the cortex. That is much more upregulated in CHI with AD, but nanowired cerebrolysin is much less, and nanowired cerebrolysin with the stem cells and antibodies to nitric oxide synthase is also significantly reduced. Next slide, please. At electron microscopy, this microglia is much more darker in appearance in CHI plus AD as compared to CHI alone. Next slide, please. Treatment with nanowired cerebrolysin help little bit to this microglia, and nanowired cerebrolysin with mesenchymal stem cells and antibodies to nitric oxide synthase. This cell markedly reduced, markedly seem normal. And this is again the same thing. We have to choose the drugs that could be better after nano delivery, and not all drugs can be better when nano delivered. Next slide, please. This is same the mechanism. They can pass the blood-brain barrier, deposit in the neuropil so that they have long neuroprotective actions. Next slide, please. As you can see that nanowired cerebrolysin with antibodies to nitric oxide synthase and mesenchymal stem cells in pathophysiology of a spinal cord injury and behavioral dysfunction won top 20% innovation award in United States in 2018. This strengthens our work to label monoclonal antibodies and stem cells with nanowired and deliver as a combined therapeutic strategy in neurological diseases. Next slide, please. This is just an example that we have been invited by Society for Neuroscience, the largest neuroscience organization in the world in the United States, to organize a session of our results with our friends and collaborators. This was in 2018. Just I'm showing you an example. Next slide, please. These are the books I showed earlier presentation. We have edited several books. Next slide, please. Yes. Drug and gene delivery to CNS for neuroprotection. Next, please. International Review of Neurobiology in 18. Progress in Brain Research 2021, I guess. Next, please. This is also in 2021, 2 volumes together. This is in 2023 about brain cancer. This is International Review of Neurobiology in 2023, I guess. Next, please. So we believe that co-administration of mesenchymal stem cells and cerebrolysin using nanowire delivery has much more superior neuroprotective effects in Alzheimer disease or in CHI, and in combination that not reported well earlier. And the basic reason is to strengthening the blood-brain barrier function in these conditions. Next slide, please. The future of nanomedicine, that we have to find comorbidity factors, how the disease can be advanced in different conditions, and then we can have successful therapy for neuroprotection that we are working on. Next slide, please. This is also the Scholar GPS. The people are highly cited scholars for different subjects. They are labeled. Next slide, please. Various organizations support our research in collaboration. Next, please. This is showing the various organizations across the world. They are involved in this huge research work together, and these are our friends across the world. I am from Uppsala University, and I really thank you, the organization, for providing an opportunity to present our results to such the prestigious organization by Mr. David. Thank you very much. Bye-bye.